Climate impact, threatening sharks with coastal upwelling. Treading the chilly depths, sharks face peril in warming seas. As the planet's climate undergoes dramatic shifts, the delicate balance of our ocean ecosystems is being disrupted in ways that threaten even the most resilient marine predators. In a captivating new study published in Nature Climate Change, researchers uncover a chilling reality facing one of the ocean's most iconic inhabitants, the mighty shark. The story begins on the southeast coast of South Africa, where in 2021, a shocking occurrence played out. Coastal communities were inundated with a startling sight: the washed-up remains of hundreds of marine creatures, spanning 81 different species of fish and invertebrates. This mass die-off coincided with a powerful shift in the Agulha current, a major offshore ocean current, which brought with it drastic changes in temperature, currents, and water chemistry. Intrigued by this phenomenon, the research team, led by Valentina dos Santos, delved deeper, uncovering a troubling connection between climate change, altered ocean circulation patterns, and the plight of one of the ocean's apex predators, the bull shark, Carcharinus lucus. Using a multidisciplinary approach that combined environmental data analysis, modeling, and animal tracking, the scientists revealed a disturbing trend. Climate-induced shifts in pressure and ocean circulation systems have intensified the frequency and severity of cold upwelling events along the western boundary currents, like the Agulha and East Australian currents. These upwelling events, where cold, deep water is brought to the surface, are proving to be deadly for marine species seeking thermal refuge. The researchers focused their investigation on the bull shark, a species known for its global distribution and relative tolerance to environmental fluctuations. By tracking the movement patterns and thermal ecology of 40 tagged bull sharks, the scientists uncovered a concerning reality. These sharks, which typically migrate between warm and cold waters to optimize their physiological processes, are now finding themselves trapped in a perilous game of hide and seek, desperately seeking refuge from the relentless onslaught of cold upwelling. The data revealed that bull sharks commonly operate near their maximum thermal thresholds when they encounter these active upwelling cells, often at the far poleward extent of their range. Forced to navigate an increasingly narrow thermal corridor, these sharks are struggling to find the conditions necessary for their survival. This study serves as a sobering reminder that the impacts of climate change extend far beyond the warming of surface waters. The cascading effects on ocean circulation patterns are creating a perfect storm for marine species, with cold upwelling events proving to be just as deadly as extreme heat waves. As the planet continues to warm, the severity of these extreme events is only set to increase, reshaping the migratory routes, movement corridors, and seasonal habitats of a multitude of marine species. For the mighty shark, the future looks uncertain, as it is forced to adapt to an ever-changing seascape, where the very elements that sustain it may one day be its undoing. Source: www.nature.com. Unraveling online polarization, exploring it as a dynamic phenomenon. Unraveling the complexities of online polarization, a dynamic perspective. In the ever-evolving digital landscape, polarization has become an increasingly complex phenomenon that defies simple explanations. Traditionally viewed as an inevitable consequence of social media use, this study takes a refreshing approach by exploring polarization as a dynamic process, shedding light on its nuances and temporal evolution. Combining expertise in political science and data analysis, the researchers delve into the polarization dynamics of two distinct debates, the COVID-19 vaccine and the Ukraine conflict, on the highly politicized platform of Twitter. Their findings challenge the notion of polarization as a static state, revealing that it is, in fact, a sequential process that varies over time and is influenced by the maturity of the debate. By analyzing the clustering of users based on their interactions and information sources, the study identifies diverse polarization patterns. Contrary to the binary view of polarized versus non-polarized users, the researchers uncover the presence of intermediate users, those who engage with both sides of the debate, yet still exhibit a preference for one community. Interestingly, the proportion of these intermediate users remains substantial, even in the face of highly polarizing debates. The true revelations, however, come when the researchers examine the temporal evolution of these polarization behaviors. Across both debates, the number and nature of user clusters fluctuate, showcasing the dynamic nature of polarization. While the COVID-19 vaccine debate exhibits a more stable progression, the Ukraine conflict debate displays a more turbulent path, with periods of unstructured, balanced, convergence, and ultimately, fully polarized user communities. Importantly, the researchers pinpoint the role of context-related events in shaping these polarization patterns. The sudden outbreak of the Ukraine conflict and the government's decision on the COVID-19 vaccination campaign trigger shifts in user behaviors, leading to the emergence of intermediate users and eventually their gradual polarization. This study's significance extends beyond merely documenting the dynamics of polarization. It paves the way for a new generation of personalized depolarization strategies, tailored to the specific context and maturity of the debates. By understanding the cyclical nature of polarization, driven by external events, the researchers suggest that diversifying information recommendations may not be a one-size-fits-all solution. Instead, interventions should be timed to coincide with the appearance of intermediate users, leveraging these periods to foster a healthier exchange of ideas. In an era where polarization is often oversimplified and treated as an immutable problem, this study stands out as a nuanced and insightful exploration of a complex social phenomenon. By approaching polarization as an evolving process, the researchers have opened up new avenues for understanding and addressing the challenges of online discourse, paving the way for a more constructive and inclusive digital landscape. Source: www.nature.com. Unlocking the phenomenon of electron bunching in quantum light, shedding new light on electron behavior, the surprising impact of quantum optics. In a groundbreaking experiment, a team of physicists has uncovered remarkable insights into the behavior of electrons when exposed to quantum light. Led by Jonas Heimel and his colleagues, this study pushes the boundaries of our understanding of light-matter interactions, revealing unexpected and intriguing phenomena. Traditionally, the photoelectric effect, where electrons are emitted from a material upon exposure to light, has been well understood when using thermal or coherent light sources. However, the interplay between quantum light and the emission of electrons has remained largely unexplored, until now. Heimel and his team generated intense pulses of squeezed quantum light and directed them onto a sharp metallic needle tip. This setup allowed them to delve deep into the nonlinear optical regime, where the ratio between the work function of the metal and the photon energy required the involvement of multiple photons to create a single photoelectron. The results were truly astonishing. Instead of the expected Poissonian distribution of emitted electrons, characteristic of classical light, the team observed a dramatically different pattern. When excited by the quantum light, the number of emitted electrons per pulse became highly unusual. Events with either zero electrons or a large number of electrons became significantly more likely than in the coherent light case. At a mean of merely 0.3 electrons per pulse, Heimel and colleagues reproducibly found events with up to 65 electrons per pulse, a result that would occur only once in more than 10 to the power of 100 repetitions in the Poissonian case, the authors write. This striking departure from the classical behavior highlights the profound impact that tailoring the quantum mechanical properties of light can have on electron dynamics. The researchers attribute this effect to the unique characteristics of the bright squeezed vacuum they employed, where the vacuum fluctuations are periodically modulated in time, leading to an oscillating uncertainty in the electric field. Interestingly, the team also observed that intentionally detuning the nonlinear optical vacuum modulator from perfect alignment caused deviations from the expected gamma distribution of electron emission, as the correlations between the even-numbered photon pairs were disrupted. In this way, one can in principle tune the electron statistics in an almost arbitrary way, as long as it stays within the constraints of quantum mechanics, the authors note. This work not only represents a significant advancement in our understanding of light-matter interactions but also opens up new avenues for potential applications. The ability to control the quantum statistics of electron emission could have implications in fields such as particle accelerators, electron microscopy, and beam-based information science. As the authors conclude, the work by Heimel and colleagues represents an enabling and groundbreaking step towards follow-up research addressing this and other questions, because it shows clearly that tailoring the quantum mechanical properties of light is not only a scientific curiosity but creates extremely strong effects that likely have practical relevance. Source: www.nature.com. Unveiling the RNA twist in high-C technology, LIMCA innovation, unraveling the genetic mysteries of the olfactory realm, a multiomics masterpiece. In a captivating tale of scientific ingenuity, we delve into the cutting-edge world of LIMCA, a revolutionary multiomics method that combines the power of single-cell genome architecture analysis with the precision of transcriptomy mapping. Crafted by the talented hands of researchers Jane Kawaoka and Stavros Lombardis, this groundbreaking technology is poised to shed light on the intricate web of genomic and transcriptomic transformations that govern the enigmatic process of olfactory receptor gene choice. The olfactory system, a veritable wonderland of sensory diversity, has long captivated the scientific community. Within the mouse main olfactory epithelium, a constant regeneration of olfactory sensory neurons, OSNs, occurs, each cell meticulously expressing a singular olfactory receptor, OR, gene from a vast repertoire of nearly 2,000 alleles. This monogenic and monoallelic expression pattern has puzzled researchers for decades, until the advent of LIMCA. Leveraging the power of nuclear cytoplasmic separation, LIMCA enables the simultaneous mapping of a cell's three-dimensional genome organization and its transcriptomy, a feat that was previously considered a formidable challenge. By subjecting the cross-linked nucleus to chromosome conformation capture and whole genome amplification, the researchers were able to achieve a staggering coverage of approximately 1 million high-C contacts per cell. Concurrently, the cytoplasmic mRNA was captured through the highly sensitive Scivi-Co protocol, providing a comprehensive view of the cell's transcriptional landscape. The true genius of LIMCA lies in its ability to unravel the intricate dance of genomic and transcriptomic events that culminate in the monogenic and monoallelic expression of olfactory receptor genes. By applying this powerful tool to the mouse olfactory epithelium, the researchers uncovered a remarkable insight. During the early stages of OSN differentiation, when multiple OR genes are co-expressed, the Greek island enhancers, key regulatory elements in the OR gene choice process, associate with the transcribed OR alleles from their own chromosomes. Over time, these enhancer hubs, or Greek island hubs, GIHs, recruit additional Greek islands from other chromosomes, converging on the co-transcribed OR alleles. Remarkably, LIMCA's unparalleled sensitivity allowed the researchers to detect subtle differences between these GIHs, revealing that the hub with the most highly transcribed OR allele exhibits more focused Greek island OR interactions compared to the hub with the second most highly transcribed OR. This observation suggests that it is the transcriptional output, rather than the sheer number of Greek islands within a hub, that ultimately determines the winner in the competition for transcriptional dominance. The LIMCA technology stands as a testament to the power of innovative multi-omic approaches, showcasing its ability to uncover the intricate mechanisms underlying cellular diversity. By seamlessly integrating genome architecture and transcriptional analysis, LIMCA has the potential to revolutionize our understanding of a wide range of biological processes, from alternative promoter activation to alternative splicing, in rare and elusive cell populations. As the scientific community continues to push the boundaries of single-cell analysis, technologies like LIMCA will undoubtedly play a pivotal role in unraveling the mysteries that have eluded us for decades. The future of biological discovery has never been more exciting, and with the advent of this multi-omics marvel, we are poised to unveil the hidden symphonies orchestrating the very fabric of life. Source: www.nature.com. Decoding the role of NCOR1/2 and glucocorticoid receptor in liver function, uncovering the orchestrators of hepatic function, a fascinating discovery. In a groundbreaking study published in Nature Metabolism, a research team led by Mitchell Lazar has unveiled a remarkable discovery about the intricate workings of the liver. The findings shed light on the unexpected and pivotal role played by the coregulators NCOR1 and NCOR2, collectively known as NCOR1/2, in orchestrating hepatic function, with far-reaching implications for our understanding of metabolic disorders. Traditionally, NCOR proteins have been associated with their repressive functions within the liver, working in tandem with the chromatin-relaxing enzyme HDAC3 to regulate lipogenic gene programs. However, this new study uncovers a previously unrecognized, HDAC3-independent and gene-activating function of NCOR1/2 that is crucial for glucose homeostasis. The researchers found that the depletion of NCOR1/2 in adult mouse livers led to a rapid onset of liver failure, marked by splenomegaly, fibrosis, steatosis, and inflammation. Surprisingly, this phenotype was more severe than that observed in single knockouts, suggesting the existence of previously unidentified transcriptional targets of NCOR1/2. Through RNA sequencing, the team uncovered a vital role for NCOR1/2 in activating genes essential for glucose regulation, a finding that was corroborated by the severe hypoglycemia exhibited by NCOR1/2 double knockout mice during fasting. Mechanistically, the researchers discovered that NCOR1/2 coordinate the regulation of chromatin accessibility, particularly at enhancer sites associated with gluconeogenesis, and are essential for the binding of the glucocorticoid receptor, GR, to the chromatin of glucoregulatory genes. This remarkable discovery challenges the long-held perception of NCOR proteins as solely repressive regulators and highlights their unexpected ability to activate gene expression programs crucial for metabolic homeostasis. The findings have far-reaching implications, not only for our understanding of the complex interplay between coregulators and nuclear receptors in liver function but also for potential therapeutic avenues. The researchers envision that this newfound understanding of the pivotal role of NCOR1/2 in regulating the fasting response could help address the undesirable hypoglycemic side effects often associated with pharmacological glucocorticoid therapy. By selectively targeting the NCOR1/2 mediated activation of gluconeogenesis, future approaches may be able to alleviate these detrimental effects and improve the management of chronic inflammatory conditions requiring long-term glucocorticoid treatment. As the field of coregulator research continues to evolve, this study by Hauk and colleagues serves as a powerful reminder of the versatility and importance of these transcriptional modulators in orchestrating metabolic pathways. The insights gained from this work open up new avenues for exploring the potential involvement of aberrant NCOR1/2 expression in human liver pathologies, such as metabolic dysfunction-associated steatotic liver disease, MASLD, and glucose dysregulation disorders. With these findings, the stage is set for further advancements in our understanding of the intricate mechanisms governing liver function and the development of innovative therapeutic strategies targeting coregulator-mediated pathways. Source: www.nature.com. Bio-inspired membrane, a natural solution for arsenic removal, diving into the depths of water purification, a remarkable breakthrough. In the ever-evolving quest to tackle the global water crisis, a team of dedicated scientists has unveiled a groundbreaking innovation that promises to revolutionize the way we purify contaminated water. Bailin Deng, a leading expert in the field of environmental engineering, has developed a remarkable bio-inspired membrane that can efficiently remove various arsenic species from polluted water sources. Arsenic contamination is a pressing environmental concern, as exposure to this toxic element can have dire consequences for human health. Deng's innovative approach draws inspiration from the remarkable arsenic-protein interactions observed in biological systems, where nature has evolved intricate mechanisms to manage this hazardous substance. The key to the membrane's success lies in its ability to mimic these natural processes, harnessing the power of specialized proteins to selectively capture and remove arsenic from the water. By delving into the intricate workings of living organisms, the researchers have uncovered a remarkable strategy that outperforms conventional water treatment methods. This bio-inspired membrane represents a significant leap forward in our efforts to combat the global water crisis, Deng explains. By tapping into the ingenious solutions nature has developed over millennia, we've created a highly efficient and environmentally friendly solution that can make a real difference in the lives of communities struggling with arsenic-contaminated water. The implications of this breakthrough are far-reaching, as the membrane's versatility extends beyond arsenic removal. It can effectively target a wide range of water pollutants, including heavy metals, organic contaminants, and even microorganisms, making it a robust and adaptable tool in the arsenal of water purification technologies. The research team's dedication and innovative thinking have paved the way for a future where clean, safe water is accessible to all. By merging cutting-edge scientific knowledge with the elegance of natural systems, they have crafted a solution that holds the promise of transforming the way we address the world's most pressing water challenges. As the scientific community and the public eagerly await the widespread adoption of this bio-inspired membrane, Deng and his team continue to explore new frontiers, driven by a relentless passion to protect the precious resource that sustains all life on our planet. Source: www.nature.com. Duplicated genes drive tissue-specific functions, unraveling the evolutionary tapestry, a tale of tissue specificity and gene duplication. In the ever-evolving world of biology, the bilaterian clade has long been a captivating subject of study. These diverse organisms, ranging from simple flatworms to complex vertebrates, possess an intricate array of organ systems and tissue types that have fascinated scientists for decades. Now, A groundbreaking study published in Nature Ecology and Evolution sheds new light on the intriguing relationship between gene duplication and the emergence of tissue-specific gene expression patterns. Anna Maria Nexilia, a talented science journalist and expert in broad scientific fields, takes us on a captivating journey through the findings of Mantica and collaborators. The researchers adopted a comprehensive approach, analyzing gene expression patterns across 8 tissue types in 20 bilaterian species, spanning both the protostem and deuterostem branches. This strategic sampling allowed them to draw insightful comparisons between evolutionary events in these distinct lineages. The study unveils a remarkable discovery. Approximately 7% of all the identified homologous gene families exhibited conserved tissue-specific expression, indicating that these core, ancestral functions were already present in the last common bilaterian ancestor. This finding speaks to the remarkable resilience and adaptability of these fundamental biological processes, as they have persisted through millions of years of evolution. But the real story lies in the dynamic patterns of tissue-specific gene expression that have emerged in the descendants of this ancient lineage. Mantica and collaborators found that tissue specificity is a highly fluid and adaptable trait, with lineage-specific gains and losses occurring throughout bilaterian evolution. This illustrates the remarkable plasticity of the ancestral gene repertoire, as conserved gene families were independently co-opted into the tissue-specific gene networks of individual species. The most intriguing aspect of this study, however, is the tight link between gene duplication and the evolution of tissue-specific expression patterns. The authors observed that the highest number of gains in tissue specificity occurred in the ancestor of vertebrates, a lineage known to have undergone 2 rounds of whole genome duplication. This finding lends strong support to Ono's seminal proposal that gene duplication is a primary driver of evolutionary novelty. Interestingly, the association between gene duplication and the emergence of tissue-specific expression was not limited to the immediate aftermath of the duplication event. The authors found that this effect persisted throughout evolutionary time, suggesting that the impact of whole genome duplications on the functional diversification of the ancestral gene repertoire may be even more profound than previously believed. As Nexilia eloquently notes, this study not only represents a valuable resource for researchers but also provides a robust conceptual framework for future investigations into the early evolution of tissue-specific gene expression. The insights gleaned from this work promise to shed new light on the intricate mechanisms that have shaped the remarkable diversity of bilaterian life. Source: www.nature.com. AI Predictions, Tackling Asia's Deadly Spring Dust Storms. Here is a 547-word story summarizing the article on the lethal dust storms that blanket Asia every spring and how AI could help predict them. Battling the Dust, How AI Aims to Tame Asia's Annual Scourge. As the warm winds of spring sweep across the Asian continent, the region braces itself for an all-too-familiar foe, the dreaded dust storms. These colossal walls of swirling sand and grit have long plagued countries from China to Pakistan, bringing with them a litany of dangers that threaten both human health and livelihoods. In late March 2024, the skies of Inner Mongolia turned a murky yellow as wind speeds reached over 60 miles per hour, reducing visibility to a mere 55 yards. Residents were urged to stay indoors, safe from the onslaught of the choking particulates. This is a scene that has played out year after year, as these dust events sweep across the region, with devastating consequences. But now, a new ally has emerged in the fight against this natural scourge: artificial intelligence. Researchers across Asia are harnessing the power of AI to help predict the timing, intensity, and movement of these dust storms, providing critical early warnings that could save lives and livelihoods. At the forefront of this effort is Chen Xiu, an atmospheric scientist at Lanzhou University in China. Chen and her team have developed a system called the Dust Watcher, which uses AI to analyze a wealth of data, from ground observations to satellite imagery, to forecast the arrival of these dust events up to 12 hours in advance. In a trial run last year, the Dust Watcher outperformed traditional models by an impressive 13% in accuracy. AI can learn how dust storms evolve in time and space from a large amount of data, explains Huang Jianping, China's leading expert in dust dynamics. And we already have a huge volume of information about dust storms, including ground-level observational data, satellite data and simulations from various models. The need for such advanced forecasting is clear. Dust storms not only reduce visibility and disrupt transportation, but they also pose a grave threat to human health. During these events, mortality from cardiovascular and respiratory diseases can spike by as much as 25% and 18%, respectively. The dust also carries bacteria and toxic particles, contaminating water sources and damaging crops. Researchers in other parts of Asia are also working to harness the power of AI and data assimilation to refine the dust storm predictions. In Nanjing, a team led by Jin Jiabing has developed the Dust Assimilation and Prediction System, DAPS, which can provide detailed 48-hour forecasts on the spread and concentration of dust across 5 countries in East Asia. While the relationship between climate change and dust storms is complex, with some regions seeing a decline in dust levels, the threat remains ever present. As Gao Meng of the Hong Kong Baptist University warns, if efforts to curb global warming are successful, West and South Asia's dust levels could rise again. The battle against this natural phenomenon is far from over, but with the aid of cutting-edge AI and data analysis, scientists are now better equipped than ever to tame the dust storms that have long plagued Asia. The stakes are high, but the potential rewards, in terms of saved lives, protected livelihoods, and a healthier environment, are immeasurable. Source: www.nature.com. Unlocking Immunotherapy Response, T-cell stroma enrichment score in urothelial carcinoma, lighting up the future, how a novel T-cell stroma score could transform urothelial cancer treatment. In the ever-evolving landscape of cancer research, a groundbreaking study has emerged that holds the promise of revolutionizing the way we approach immunotherapy for urothelial carcinoma. Led by a team of esteemed scientists, this study has uncovered a novel biomarker that could pave the way for more personalized and effective cancer treatments. The study, published in the prestigious Nature Reviews Clinical Oncology, delves into the intricacies of the tumor microenvironment and its pivotal role in shaping the immune response to cancer. Traditionally, researchers have focused on PD-L1 expression as a predictor of immunotherapy response, but the results have been inconsistent, plagued by issues such as spatial and temporal heterogeneity. Recognizing the need for a more robust and reliable biomarker, the researchers employed a multi-omic approach, combining whole genome sequencing and RNA sequencing, to define a transcriptomic immune-based biomarker that could guide the use of immune checkpoint inhibitors, ICIs, in urothelial carcinoma. The cornerstone of their findings is the development of a T-cell to stroma enrichment, TC, score, a gene expression-based metric that quantifies the balance between T-cell infiltration and stromal cell involvement within the tumor. Remarkably, the researchers found that tumors with positive TC scores, indicative of a T-cell-rich and stroma-poor microenvironment, were associated with superior response rates and improved progression-free and overall survival in patients treated with pembrolizumab. Importantly, the TC score outperformed other established biomarkers, such as tumor mutational burden and APOBEC mutagenesis, in predicting ICI response. The researchers validated their findings in larger datasets, further solidifying the potential of this novel biomarker. The implications of this study extend beyond the realm of urothelial carcinoma. As the authors note, the TC score may have broader applicability to ICI-containing combination regimens and other emerging therapies, such as antibody-drug conjugates and chemotherapy. This versatility could unlock new avenues for personalized cancer care, where clinicians can tailor treatment strategies based on the unique characteristics of each patient's tumor. While challenges remain in the standardization and practical implementation of RNA sequencing-based biomarkers, the insights from this study provide a strong foundation for future research. As the authors suggest, the development of more accessible, immunohistochemistry-based surrogates could further facilitate the clinical adoption of the TC score. In the ever-evolving landscape of cancer research, this study stands as a shining example of the power of multi-omics approaches to uncover novel biomarkers that can transform the way we treat cancer. By shedding light on the complex interplay between the immune system and the tumor microenvironment, the researchers have ignited a new era of personalized cancer care, where the torch of hope burns brighter than ever before. Source: www.nature.com. Unlocking Cosmology's Greatest Mystery, Can JWST Solve the Universe's Expansion Debate? Unraveling the Cosmic Mystery, the debate over the universe's expansion rate. In the rapidly evolving field of cosmology, one of the most fundamental questions has remained elusive: just how fast is the universe expanding? The long-standing discrepancy between observations of the current universe and predictions based on early universe data has left cosmologists puzzled, and they are now turning to the powerful James Webb Space Telescope, JWST, to help solve this cosmic conundrum. The latest findings from two teams of cosmologists, presented at a recent conference in London, have only further complicated the picture. Wendy Friedman's team, using JWST observations, calculated a Hubble constant, the measure of the universe's expansion rate, of 69.1 km per second per megaparsec, which is only slightly higher than the 67 km per second per megaparsec predicted by the Planck satellite's early universe data. However, this is at odds with the work of Adam Rees and his collaborators, who measured a substantially higher Hubble constant of at least 73 km per second per megaparsec. The battle lines have been drawn, and the stakes are high. Reconciling these divergent results could hold the key to unlocking a deeper understanding of the fundamental nature of the universe and the physics that govern its evolution. As Friedman aptly stated, we need more than one method, and we need more than three if we want to put this issue to rest. Cosmologists are exploring various avenues to crack this cosmic conundrum. Kristen McQuinn, leading her own JWST study on standard candles, the bright celestial objects used to measure cosmic distances, cautions against drawing premature conclusions, emphasizing the need to thoroughly evaluate the data before making any definitive statements. Meanwhile, theorists have been working overtime to find explanations for the Hubble constant discrepancy, but as cosmologist Eleonora De Valentino notes, at least 500 models have been proposed, and none of them is satisfactory. The possibility of the current theoretical model, based on Einstein's general theory of relativity, needing to be amended looms large. Amidst the uncertainty, some cosmologists, such as George F. Stathiou and Haranya Peiris, see a glimmer of hope. They note that the latest JWST results are remarkably close to the Planck predictions, and they suggest that the incorporation of a completely new technique, such as observations of gravitational waves, could provide the missing piece to the puzzle. As the scientific community eagerly awaits the resolution of this long-standing conundrum, one thing is certain: the quest to unravel the secrets of the universe's expansion rate continues, and the JWST, with its unprecedented capabilities, may hold the key to unlocking this cosmic mystery. Source: www.nature.com. Redefining human-AI competition, evolution of benchmarks urgently required, captivating revelations, how AI is reshaping the scientific landscape. In a remarkable turn of events, the scientific community has been left awestruck by the unprecedented capabilities of artificial intelligence, AI. A groundbreaking report has revealed that these advanced algorithms are now outperforming humans in a wide range of basic tasks, challenging the long-standing belief that certain cognitive abilities were the exclusive domain of the human mind. The report, meticulously crafted by a team of leading experts, delves into the rapidly evolving landscape of AI and its profound implications for the future of science. The findings are nothing short of astonishing, as they uncover the startling proficiency of AI in areas once considered the sole purview of human experts. One particularly striking revelation is the ability of AI language models, such as the renowned ChatGPT, to produce abstracts that can fool even seasoned scientists. These AI-generated summaries, indistinguishable from those written by human authors, have raised eyebrows and sparked a crucial debate within the scientific community. The implications of this discovery are far-reaching, explains Dr. Emilia Alonso, a renowned computational biologist. We must now grapple with the realization that the very foundation of scientific discourse, the peer review process, could be vulnerable to AI infiltration. This calls for a fundamental rethinking of our evaluation and validation mechanisms. Beyond the challenges posed by AI-generated content, the report also delves into the broader transformative power of these technologies within the realm of scientific research. From drug discovery to energy optimization, AI is demonstrating its versatility in tackling complex problems that were once the exclusive domain of human expertise. The rise of AI in science is akin to a seismic shift, says Dr. Liam Nguyen, a leading expert in computational chemistry. It's not just about automating tasks or enhancing productivity, it's about unlocking new frontiers of knowledge and redefining the very boundaries of what's possible in scientific inquiry. As the scientific community grapples with these rapid advancements, the report emphasizes the urgent need to establish robust benchmarks and evaluation frameworks that can keep pace with the ever-evolving capabilities of AI. This will not only ensure the integrity of scientific processes but also pave the way for a more collaborative and synergistic relationship between human researchers and their AI counterparts. In a world where the line between human and machine intelligence continues to blur, this report serves as a wake-up call, urging the scientific community to embrace the transformative power of AI while also remaining vigilant and proactive in shaping its future impact. Source: www.nature.com. Enhancing Image Recognition with Advanced DenseNet Algorithm, Unveiling the Power of Improved DenseNet, a Transformative Approach to Accurate Image Recognition. In the ever-evolving world of artificial intelligence, the quest for enhanced image recognition technology has become a critical pursuit. Researchers have been diligently tackling the challenges posed by existing image recognition models, seeking to improve their accuracy, efficiency, and versatility. Enter the groundbreaking work of a team of talented scientists, who have developed a novel approach that takes image recognition to new heights. Leveraging the strengths of the dense convolutional network, DenseNet, architecture, the researchers have introduced a series of ingenious improvements that have the potential to revolutionize the field. At the heart of their innovation lies a two-pronged strategy. First, the researchers have optimized the feature reuse method within the DenseNet, effectively reducing the model's complexity and training costs. By adjusting the growth rate and network width, they have streamlined the model, ensuring efficient parameter utilization without compromising the recognition accuracy. Secondly, the researchers have tackled the long-standing challenge of communication bottlenecks in parallel training algorithms. Drawing inspiration from gradient quantization techniques, they have devised a novel data parallel algorithm that seamlessly overlaps computation and communication processes. This breakthrough not only accelerates the training speed but also addresses the challenges posed by large-scale datasets and complex models. The results of their extensive testing are nothing short of remarkable. The improved DenseNet models, dubbed DENSENET 50, DENSENET 100, and DENSENET 200, have demonstrated remarkable recognition accuracy, with maximum values of 92.3%, 95.4%, and 97.2%, respectively. Notably, the DENSENET 100 outperformed the renowned VGG and efficient net models across a diverse range of datasets, showcasing its robust and versatile performance. Moreover, the researchers' parallel acceleration algorithm, enhanced by gradient quantization, has proven to be a game-changer. Compared to traditional synchronous data parallel and stale synchronous parallel algorithms, the optimized approach achieved a remarkable acceleration ratio, with a maximum improvement of 1.92. This not only ensures faster training speeds but also effectively addresses the communication bottleneck challenge, paving the way for more efficient and scalable image recognition solutions. The implications of this study extend far beyond the academic realm. The improved DenseNet model and the parallel acceleration algorithm have the potential to unlock new frontiers in various industries, from computer vision and medical diagnostics to autonomous vehicles and intelligent security systems. By enhancing the accuracy and efficiency of image recognition, these innovations hold the promise of transforming the way we interact with and leverage visual data. As the scientific community continues to push the boundaries of artificial intelligence, the work of this dedicated team stands as a testament to the power of innovative thinking and relentless pursuit of excellence. Their contributions not only enrich the theoretical foundations of dense convolutional networks and parallel computing but also pave the way for a future where image recognition technology becomes an even more indispensable tool in our digitally driven world. Source: www.nature.com